Hello, and thank you for inviting me to this conference. I'm Federica, and I'm the lead biocurator of DisProt, a database of manually curated annotations of intrinsically disordered proteins and regions from literature. So in 2020, DisProt transitioned to a more frequent 6-month based release schedule, and it also underwent several improvements. In the sense that we have been focusing not only on adding new entries of IDPs and ID regions, but also revising and updating already existing entries. So we want to continuously improve the curation accuracy and provide comprehensive and up-to-date annotations for our database. And this can be done thanks to the effort of a small team of expert DisProt curators, which are our reviewers. So what we started doing in 2020 is annotating intrinsically disordered regions from SARS-CoV-2 virus as a response to the global pandemic. Indeed, we have been able to add, for example, the spike-like protein entry and or 3A protein entry. But we have also been reviewing and updating entries of well-known IDPs in light of new results that have been published in literature, so to provide always the most comprehensive and up-to-date annotation for our proteins. For example, we have been revising P27, which is a protein that plays a crucial role in cancer. And then we introduced a relevant new feature, which is the thematic datasets of IDPs, meaning that we are now exploring several biological areas where IDPs play a crucial role. For example, the first dataset described the unicellular toxin and antitoxins, and this year we have already been releasing a dataset of extracellular matrix proteins. So both of them are accessible from the DisProt homepage. Then in order to improve interoperability and data standardization of our annotations, we have been adopting two stable ontologies, the gene ontology for the annotation of interaction partners and functions of IDPs and ID regions, and the evidence and conclusion ontology for the annotation of the methods, the experimental methods used to characterize intrinsic disorder in proteins. We have been able in the last year and a half to release more than 450 new entries and more than 2,000 new pieces of evidence. So the next steps in the database are to explore new thematic datasets of IDPs and other areas where these proteins are, play a crucial role, for example, cancer, transcription factors, and viruses, but also improving and the existing entries that we have in the database, along with adding new protein entries. Finally, in DisProt, we strongly believe in training, in curation training. So one of our major efforts is always to provide new training and new curation material for our curators. And if you're interested in DisProt, these are the two main references. You can look for the NAR paper describing the database and a protocol describing how to use it. And if you want to explore the database, you can look for it here. And here we have the Twitter accounts of the database. So I would like to thank our reviewers, Lucia, Tamas, Attila, Balint, Matthias, and Rita for their amazing effort, and you for attending this talk.